Good afternoon. I would like to thank NFSTC for inviting me to speak today, and the National Institute of Justice for providing the funding that allowed us to do this research. I also thank the staff at NFSTC for the general assistance. This work was supported by an award from the Office of Justice Programs, National Institute of Justice, Department of Justice. Points of view in this document are those of the authors and do not necessarily represent the official position of the U.S. Department of Justice. In the light of tight fiscal times, the stress on the laboratory to maintain quality while producing more results will surely increase as prosecutors and police seek to solve more crimes with DNA. The question may be asked, how can the lab maintain the balance between meeting the needs of the criminal justice community while using precious resources wisely? Most of us have experienced crime scenes which contain bloodstains from multiple donors located throughout the scene. The scene may be where a drug deal went bad to a love triangle that involves both men and women. The scene has blood from individuals who were injured and continue to bleed during the course of the event. The number of stains is overwhelming, but yet there is a need to test the probative samples. The crime scene technicians are willing to collect most of the stains. The prosecutor feels that if the evidence is collected, it must be analyzed, and the laboratory must sort through the submitted evidence. Discussions occur between the laboratory, the police, and the prosecutor. Where should the laboratory efforts be placed, and what samples must be analyzed to most benefit the adjudication of the case? Those discussions would focus on, what does the prosecutor really want? Would she be satisfied with finding blood that was transferred from the victim to the suspect's clothing, and vice versa? If we have a limited population of blood donors, at what level of significance does the analytical results need to be? That is, can the samples be sorted into groups of possible contributors? By sorting the stains into possible donors, can the laboratory meet the needs of the criminal justice community by only fully typing those samples that warrant further analysis? Sample individualization is routinely performed using STR profiling with conventional capillary electrophoresis methods. However, it is often impractical to use such costly and labor-intensive methods to analyze each and every sample found at a crime scene. A simple analytical method, which could allow the analyst to select only those probative samples for further analysis, would save time and resources. With this as a backdrop, one could ask the following analytical questions. Given a limited population set of donors, how definitive does the assay need to be to sort the samples? That is, if we understand who the possible contributors to the crime scene are, can we sort those stains into those who may be donors? For example, can we screen victims' clothing for suspect blood in the midst of victims' blood? What assay could provide a screening of the blood stains to allow a determination of which stains require further testing? Many of you may remember the early days of DNA analysis, which overlapped with conventional serological typing of biological stains. Many laboratories use conventional serological typing to help sort stains prior to RFLP analysis. This allowed analysts to decide which stains may be probative prior to moving forward with DNA typing. This serological approach to sorting stains was discontinued due to many factors. However, given the wide variety of molecular biological testing methodologies currently available, could we employ one of these methods to sort stains? In order for this assay to be useful for this approach, it must be rapid, use a limited amount of sample, provide enough statistical weight to allow the samples from the crime scene to be associated with one of the bleeders, use existing forensic lab instrumentation, maximize the information obtained from the sample, not be too expensive, and provide an indication of a mixture. Prior to building an assay, we should review the assays that are available today. Currently, quantitation kits are available that allow us to determine if a stain has been donated by a male or female individual. These tests are rapid, use limited sample, and can quickly sort stains into two groups. However, if the crime scene requires more than just this type of sorting, this simple assay will not be sufficient for our work. Considerable work has been conducted on SNP-based assays that provide some information about the donor's ethnicity or phenotypic characteristics. These tests may be useful in some circumstances where the suspects are unknown, and information relevant to the investigator to identify potential suspects would be useful. However, if one just wants to sort stains into possible donors, a test designed for that purpose may be more appropriate. In designing our research to solve this problem, we decided that the test need not be definitive in nature. The assay did not have to yield a complete profile concerning the donor of a particular stain, but reveal sufficient data to allow the examiner to differentiate between those individuals present at the crime scene. Only stains which yielded information that indicated a need for further examination would be tested using conventional methods, thereby eliminating many stains which would provide no useful purpose if subjected to lengthy testing. A method to quickly type only a few informative loci, while certainly not by any means a definitive test of identity, could allow determination of whether a blood sample came from a particular contributor at a crime scene. With such information, one could screen crime scene samples to determine the most probative samples. With this in mind, we felt that SNPs could provide a limited statistical assessment of samples to provide the ability to sort stains from a limited population of donors. There are numerous approaches to the analysis of SNPs. A few common methods are noted here. The single nucleotide extension method can be purchased from Applied Biosystems. Their kit is known as the SNaPshot Primer Extension Assay. Applied Biosystems also offers a TaqMan real-time PCR assay. Luminex provides a bead array with allele-specific hybridization. Pyrosequencing is another method, and fluorescence resonance energy transfer, FRET, hybridization probes is another SNP analysis assay example. The SNaPshot Primer Extension Assay uses labeled dideoxynucleotide triphosphates in conjunction with primers that sit adjacent to the SNP of interest. The polymerase incorporates the labeled dideoxynucleotide, and the polymerization is halted. The labeled nucleotide is complementary to the SNP base. The denatured primer with labeled nucleotide is analyzed via a genetic analyzer, and the labeled base is interpreted to determine the complementary base. As this diagram illustrates, DNA template is added to the SNaPshot reaction mix with the appropriate primer. The primer is designed to be adjacent to the target nucleotide of interest, which in this case is a G. During amplification, the labeled C is incorporated. Later, after removal of the unused nucleotides, the product is denatured and electrophoresis is performed on a genetic analyzer to determine which base was added to the primer. This is a powerful technology, but it requires the examiner to produce PCR product to serve as a DNA template, perform the SNaPshot reaction, manipulate the amplified product, and run the product on a genetic analyzer. This is time-consuming and ties up the CE for screening-type assays. The TaqMan real-time PCR assays by Applied Biosystems are very useful for SNP analysis, but this method requires 2 labeled probes for each SNP locus, which places a limitation on multiplexing, as a 4-color instrument can only yield results for 2 SNPs. This slide illustrates the TaqMan approach to SNP analysis. This is a typical TaqMan procedure, except that 2 probes are included in the assay. Each probe is selective for a particular SNP, with a dye to indicate which probe bound to the target region. In this example, if the template has a C present at the SNP location, the probe with a G base in the complementary location will bind and the VIC dye will be released and become fluorescent. Likewise, if the A probe binds the template where the C is substituted for a T, the FAM dye will be released with subsequent fluorescence. There are other popular SNP-based assays. Luminex bead array with allele-specific hybridization is one method, and pyrosequencing is another. Both Luminex and pyrosequencing require instruments not typically available in a forensic laboratory. Another approach to this problem is the use of FRET hybridization probes. In this procedure, dual probes are used with a subsequent melt curve analysis. This process uses 1 fluorescent probe per locus. In reviewing dual probe methodology, we asked if real-time PCR in conjunction with this technique could allow us to develop efficient methods to quickly sort stains and possibly identify a sample as coming from a victim or possible suspect. Compared to other technologies, a real-time PCR approach provides an instrument available to most forensic laboratories, typically quick assays with multiple samples analyzed simultaneously. Limited amount of sample is required, and the approach doesn't tie up the genetic analyzer. This analysis, which we call the stain donor assay, is SNP-based, using a real-time PCR instrument. Incorporates a multiplex assay, which is FRIT-based using dual probes. One color provides information about both alleles in a locus through a melt curve analysis. With appropriate standards, one could determine quantitative information about the extracted sample, such that this procedure could replace the traditional real-time PCR quantitation, and also quickly provides a sorting of the stains found at a crime scene. This assay was published in the Journal of Forensic Sciences in 2008. SNPs employed in this assay were selected from the Alfred database, established by Ken Kidd of Yale, with a selection of SNPs such that the variation in allele frequencies between populations were low, that is, loci with a low FST. We did not want these SNPs to be predictive of ethnicity, but rather good at differentiating individuals. Non-coding and not medically relevant, allele frequencies close to 0.5 to provide the most information from a particular locus, located on different chromosomes, and not patented. There are several key aspects of the FRET dual probe hybridization assay. The hybridization assay is based on two probes binding to the region of interest. The first probe is the SNP probe, or sensor probe. This probe is a perfect complement to one SNP allele, and hence one base mismatch to the other allele. In addition, the sensor probe contains a 3' fluorophore. In a multiplex, additional sensor probes would be designed for additional SNP sites with different fluorescent dyes. The second probe in the assay is known as the anchor probe. The anchor probe binds adjacent to the SNP probe on the DNA template. This anchor probe contains a 5' quencher, which quenches the sensor probe fluorophore when both probes are bound to the target. The anchor is designed to have a higher TM than the sensor probe. This melting temperature difference is important, and for the assay to work, the anchor probe must have a higher TM than the sensor probe so that the melting profile is dependent upon the base mismatch of the sensor probe. The assay is conducted in two steps. First, PCR is performed and product is produced. Monitoring the change in fluorescence can be done if quantitation of the sample is desired. After PCR, the melt step is conducted. The PCR tube is left closed, and the double-stranded DNA amplicon is melted. The temperature is reduced to re-anneal the hybridization probes. The temperature is then slowly increased, and fluorescence is monitored as the probes melt. This melt data can be converted to easily interpretable data with software that plots the negative first derivative of the melt curve data. This slide diagrammatically details the components of the assay. For each SNP under analysis, there are forward and reverse primer, a sensor and anchor probe, and template in addition to the master mix. PCR is conducted, and the fluorescence can be monitored to review if amplification took place and if quantitation of the extracted sample is desired. This diagram shows the initial PCR process and the decrease in fluorescence over time as the probes bind to an increasing amount of PCR product. After PCR, the double-stranded amplicon is melted, the temperature is reduced, and the hybridization probes are re-annealed. The temperature is slowly increased, and the fluorescence is monitored to note the temperature at which the probe melts from the template. The melt analysis software allows the examiner to quickly review the differences in melting points, which reflects the binding differences between the probe and the template. At temperatures below the TM for the sensor probe, the probe binds to both matching and mismatching sequences. The anchor probe binds adjacent to the sensor probe, quenching the fluorescence from the sensor probe. In the following example, the sensor probe is a perfect match for the sequence with a SNP-A allele. In this example, below 61 degrees, the fluorescence remains constant. The cartoon in the lower right shows that during this temperature range, both sensor probes, the one that matches perfectly and the mismatching probe, remain bound to the template. As the temperature is raised, the mismatching probe begins to melt, leaving the perfectly matched probe still bound to the template. When the mismatching probe melts, the distance between the fluorophore and the quencher increases, and the fluorescence from this probe is now detected. As this graph demonstrates, as the temperature is slowly raised above 67 degrees, the matching probe melts with an increase in fluorescence, as noted on the graph. The cartoon on the lower right of the slide shows both probes are unbound and in solution at this temperature. At this point, the data obtained from the melting of the probes is now interpreted by the software, which plots the negative first derivative to allow the examiner to easily interpret the data representing the different alleles. This slide shows the results of the software analysis on the melt curve data. The blue curve shows a sample that is a AA homozygote. In our example, the matching probe contained a T. This sensor probe melted around 69 degrees, whereas the mismatching probe melted at a lower temperature at about 62 degrees, as indicated by the yellow curve for the homozygote CC. The red and green lines show what a heterozygote would look like. This slide shows the melt curve results for the FLJ locus, one of the loci in our published multiplex. The green curves represent samples that are heterozygous for the genotype AC. The red curves are the homozygote samples AA, and the blue curve shows the results for a CC sample. We have developed and published 2 quadruplex assays and 1 hexaplex for 6-color instruments. The next 2 slides show the results of other loci in their color channels noted as green, yellow, and crimson and blue. Included on each graph are curves showing samples that are homozygous and heterozygous for each locus. There is considerable data on this slide. However, in essence, we conducted a sensitivity study, and at a concentration of 72 picograms, all homozygous samples were detected. However, the assay begins to not detect all expected heterozygous alleles at this concentration. For screening purposes, this assay could be very useful to sort stains found at a crime scene. Using an assay where both alleles are represented in the general population at about 50%, that is, where P equals Q equals 0.5, the probability of 2 random individuals having the same result, that is, the same genotype, is 2% for a 4 SNP assay, 0.3% for a 6 SNP assay, and about 1 in 2,500 for an 8 SNP assay. Although most labs have only a 4-color instrument, standards from the individuals involved could be analyzed initially, using the 2 quadruplexes to see which assay would be most useful to differentiate the individuals prior to running the crime scene samples. In the developed assays, the primers are up to 25 bases long, and the SNP products range from 141 base pairs to 192 base pairs in length. The sex determinant product has a 1 nucleotide difference between the ZFX and ZFY genes and is 245 base pairs in length. The PCR specifications for this assay are shown on this slide. The PCR is 45 cycles using typical PCR parameters. The difference comes at the end. The last cycle melts the double-stranded DNA and reduces the temperature to allow the hybridization probes to reanneal. Afterwards, the melt phase is carried out from 50 degrees to 80 degrees C by increasing the temperature by 1 degree steps and waiting for 5 seconds at each step. As discussed earlier, the assay can also be used to measure concentration of the extracted samples. Similar to how Promega's Plexar assay works, the fluorescence decreases as PCR product increases. Standard curves can be run and quantitation performed similarly to what most laboratories do today. This slide shows the quantitation results of 2 quadruplex assays. The values in the bottom table represents the calculated concentrations based upon the standard curve results using that locus. 2 loci are highlighted for an example. The initial results employing the assay for determining sample concentrations are promising. The R-squared in this example range from 0.998 to 0.974. One could take the mean of the concentration from all assays or use a locus which provides the best R-squared. Results compared favorably to those results we obtained with Vermont's published ALU-based assay. More work is required before this could be implemented routinely. Let's take a moment to do a quick review of the assay. The assay provides a sorting of the stains. It is relatively quick, requires a limited amount of sample, not labor-intensive, easy to interpret, and provides quantitation results of the extracted sample. However, there are some issues with this method. Since the assay only reveals at most 2 alleles at each locus, mixtures may be hard to detect and may be missed at low minor contributor concentrations. For this reason, protocols may want to include the STR analysis of those samples that appear to be mixtures. Another issue with this method is one of in-house production of the assay. Although this SNP assay uses less primers and probes per locus than the TaqMan approach, it still requires 2 primers and probes for each locus. And for a 4-locus assay, you need a combination of 16 primers and probes. This assay is fairly labor-intensive to produce, and one may ask if the average forensic laboratory would want to construct this assay. Or is there an easier way? The melt curve analysis of SNPs proved very interesting, but somewhat complicated to produce as an assay. We wanted to see if we could do something similar and perhaps simpler by melting STRs. So we asked the following questions. Could we develop a simple assay where selected STRs were amplified and melted to provide differentiable melt curves? Could we use high-resolution melting to evaluate a few STR loci to provide discrimination of stains in a small population set? The approach was quite simple. All we need is 2 primers for each locus and intercalating dye specific for the melt. High-resolution melting is used to study the thermal denaturation of double-stranded DNA and has been successfully used in a number of applications. PCR products can be differentiated based on length, sequence, and single base changes, such as those found in mutations and SNPs. As STRs are length-based and have sequence differences, the HRM approach should work if the appropriate STRs are selected. The HRM method requires 2 amplification primers, a PCR master mix containing a saturating intercalating dye that binds specifically to double-stranded DNA but negligibly to single-stranded DNA. This dye must not be an inhibitor to DNA amplification, and a real-time PCR instrument that provides a high-resolution melt. The goal of this project was to develop an assay that was simple and fast and provided sufficient information to differentiate between samples originating from different donors. The goal was not to unequivocally identify STR types. This work will be published in a future publication in the Journal of Forensic Science early in 2012. Briefly, PCR is conducted in a fashion similar to routine PCR. After the product is produced, the high-resolution melting takes place and the change in fluorescence is monitored. The data can be interpreted by software, although we found more success by reviewing the differences in melt curve data by eye. The method we developed involves 3 singleplex assays. 3 separate amplifications are performed, 1 for each locus, followed by a melt of the amplified product. The results for 3 loci are depicted here, placed on the same graph for comparison purposes. The data show 3 amplification and melts for the same sample and depicts the different melt curves for the 3 loci. This slide shows melt curve results for 5 different samples. 5 different samples were amplified with VWA primers and then subjected to melt curve analysis. As can be seen, the melt profiles are different for each of these samples. 2 different DNA samples were subjected to amplification and melt curve analysis. This graph combines melt curve results from 3 loci, showing that the melt profiles are quite different for these samples. The gray line represents sample 1, the black sample 2. Sequence variations can be revealed with high-resolution melt analysis, and STR samples with the same STR genotype can have different melting profiles due to these internal variations. This graph shows the melt pattern for 5 DNA samples with the genotype 16-17 for VWA. 2 samples show one melting pattern as detailed by the black curve, while the other 3 show a clearly different pattern in the gray curves. Sequencing analysis of these samples was conducted, and sequence variations between the gray and black curve samples were observed. Initial quantitation results indicate that this approach could be coupled to a standard curve to yield both quantitation data and information useful to sort stains. This presentation has demonstrated the possibility of using melt curve analysis to assist in sorting crime scene samples. The procedures allow for selection of suitable samples for downstream analysis without wasting valuable sample or resources. The integration of such an approach may become useful for cases where numerous biological stains require analysis. I'd like to thank those at the Vermont Forensic Laboratory, especially Jan Nicholas and Tricia Conti, for their work on this project, and the National Institute of Justice for funding and support. Thank you for your attention, and please feel free to contact me for protocols or additional information at the email address above.